They're looking for one tiny clue, the one genetic mutation that matters, but it's buried under a literal mountain of false leads. They're trying to find a signal in an ocean of noise. Well, today, we're going to look at how AI is finally giving us a way to tune out all that static and clearly hear the message that could save lives. Sounds a little weird, right? But what if I told you it was basically true? For decades, hospitals have been saving millions of patient tissue samples, creating this unbelievable library of diseases. But there's a catch, and it's a huge one that's been holding back cancer research for years. So let's talk about this library of disease. For decades, every time a tumor was removed, a tiny piece was carefully saved. We're talking about millions and millions of samples all sitting in storage. Together, they make up this incredible historical record of cancer, a potential goldmine of information just waiting for us to figure out how to read it. And this is how they did it. It's a standard method called FFPE. Basically, they fix the tissues in a chemical, formalin, and then they embed them in paraffin wax. And look, it's fantastic for storing these samples for a long, long time. But it does something pretty awful to the DNA locked inside. And that right there is the central problem. The very chemical that preserves the tissue, it also wreaks havoc on the DNA. It causes all these chemical reactions that create thousands of false genetic mutations. This right here is our noise. And just to give you an idea of the scale of this problem, take a look at this chart. It's kind of mind-blowing. See the red bars? That's the noise. That's the fake stuff. In some of these cancer samples, more than 98% of the mutations they find are just errors from the preservation. I mean, can you imagine? For every 100 clues, 98 of them are completely fake? So how in the world do you tell them apart? Well, luckily, the noise, the artifacts, it has a kind of signature. These fake mutations, they tend to show up in a tiny fraction of the DNA reads, what's called a low allele frequency. And they often appear on only one strand of the DNA, which gives them a high strand bias. The real mutations, the signal we're looking for, they just don't usually act like that. And this graph just lays it all out for us. It's so clear. On the left, you've got the allele frequency. See how the red boxes, the artifacts, are all squished down at the very bottom? And then on the right, the strand bias score. The red boxes are way up high. The patterns are definitely there, but you see that tricky little bit of overlap? That's what makes just using a simple filter so incredibly hard. And that brings us to the core issue. Because the noise and the signal kind of bleed into each other, the old methods left scientists with this impossible choice. Either you leave the data so noisy it's practically useless, or you apply a filter that's so harsh you risk throwing away the actual life-saving clues you were looking for in the first place. This is where the stakes get so high. The researchers put it perfectly. Crude filters risk throwing out what they call true subclonal mutations. Okay, so what are those? They're real mutations, but they only exist in a small fraction of the tumor cells. They might seem small, but they can be absolutely critical for understanding how a cancer might fight back against treatment. Losing them is just not an option. So let's look at the old playbook. You had the MuTech filter, which barely made a dent. It left most of the noise right where it was. Then you had another tool, FFPolish. It was better at cleaning, but it had a fatal flaw. It threw out all of those super important low-frequency true mutations. Every single one. So you were completely stuck. So this is where a totally new approach was needed. Instead of trying to use a crude filter, what if you could train a highly intelligent detective to go in and examine every single piece of evidence one by one? And that is exactly what these researchers did. They built a brand new AI-powered tool, and they called it Deep Pomics FFPE. It's built on something called a deep neural network, and it's basically the expert genetic detective that this field has been desperately waiting for. The easiest way to think about it is like a digital brain. You don't just give it a bunch of simple if-then rules. No. Instead, you show it thousands and thousands of examples, and it just learns. It teaches itself to see the really subtle, complex patterns that separate the real thing from the fakes. So here's how they did it, and it's pretty clever. They took data from tumors that were both fresh frozen, so that's the clean signal, and the matching preserved sample, the super noisy version. They had the AI analyze 41 different features for every potential mutation, and over time, it learned the unique fingerprint of a preservation error versus a real genetic variant. Okay, so our AI detective has graduated from training. Now it's time for the real test. How does it actually stack up against those old, clunky methods? Well, the results are, honestly, they're just staggering. Let's break this table down. Look at that first column, specificity. That's how many artifacts it removed. Deep Omics FPF FOJA hits 99.6%. It gets rid of virtually all the junk. But here's the crucial part. Look at the next column, sensitivity, or true variance kept. It still holds on to over 87% of the real stuff. That F1 score at the end just measures the overall balance. And at 88.3%, it doesn't just beat the other tools, it completely blows them out of the water. Let's just park on this number for a second. 99.6%. It successfully found and removed pretty much every single piece of noise from the data. That is just an incredible level of precision. And here is the other half of the magic trick. It did all of that cleanup while still correctly keeping 87% of the true genetic mutations. It finally found that perfect balance that was impossible before, getting rid of the noise without losing the precious signal. But here, this is the real killer test. Remember those super important low-frequency mutations we talked about? The ones the old filters just couldn't handle? Well, look at this. FFPolish had a sensitivity of 0. It missed all of them. But Deep Omics FFPE, it successfully finds these incredibly faint signals while still getting rid of almost all the noise. It can finally see what was completely invisible before. So what does this all mean in the real world for actual patients? Because this isn't just some academic exercise in cleaning up data. This has a direct line to the future of how we treat cancer. And why this matters is actually pretty simple. With clean, reliable genetic data from all these archived tissues, doctors can suddenly get a crystal clear picture of a patient's unique cancer. This is the raw material you need to design personalized cancer vaccines or to create custom blood tests that can track if a treatment is working in real time. It's a game changer. So when you step back and look at the big picture, what's happening here is really profound. We're not just cleaning up some files. We are unlocking a vast library that contains decades of human experience with cancer. This AI, it's the key that lets us finally read these incredibly important books that have been sitting on the shelf, unreadable for way too long. It really makes you wonder, doesn't it? We just watched an AI solve a massive data problem that was a huge roadblock for personalized medicine. So,